Okay, so thank you everybody for being here. I'm Damien. I'm a biological modeler. I will try to be precise, but I try also to explain to the broader what we want to do. And so if it's not clear, feel free to shoot. But first of all, I would like to thank all the co-authors of this study. And first of all, Antoine Regimbaud, because I will just present the framework basically of his PhD thesis. In particular, so you could read all the co-authors. It's a long process. It's very interdisciplinary, and but anyway, the full credit belongs to Antoine. What I propose to do is to bring you to the footsteps of Jacques Monod. So I don't know if you guys are aware about who is Jacques Monod. So to me, it's like it's like a god. He did many things. So he's known mostly for oceanographers for the Monod law, for the way to represent microbes, the way to represent ordinary differential equations that represent the growth rate of microbes. It's also well known for making a new machine called a chemostat. That's something that allows you to culture bacteria. This is a machine that is already that he proposed to test several hypotheses. And actually, it is a machine that is still used today in most of microbial laboratories. But most importantly, is also the Nobel Prize of medicine, because he's the guy that proposed the concept of genes and how the genes are talking to each other. So I like to start with Jacques Monod because actually he's one of the few biologists that actually propose a new concept, which is the concept of genes and the concept of operons. So operons, this is units of operations that genes allows genes to talk to each other. He was proposing new formalisms, new equations that are still used today. I will present it a bit later. But he just proposed a new revolution. And if today we are talking about genetics, if we are talking about genes, this is actually from Jacques Monod. So we know the genes from Mendel, I mean, this kind of theory, but the name gene is actually a French name, gène, associated to constraints. And this is something important because I will present it a bit, something called constraints programming. So that's why I like to say that maybe we are following Jacques Monod footsteps. And following Jacques Monod, there was an emergence of a field called systems biology. So I know that most of you guys are not necessarily acclimated to biology, but you have to acknowledge that it has been the emergence of two different fields. One field is represented here in blue, basically new concept like genetics, DNA, all this kind of stuff that maybe you heard about from the news. So this is high throughput sequencing, the fact that you could now get the genome from humans, and more recently, what is something called the metagenome. So we could extract all the genes that belong from one given environment. But we have to acknowledge that at the same time, there was an evolution as well over time about all the concepts that are very useful in biology. So non-equilibrium thermodynamics, the concept of self-organizations, the concept of operon genes. So again, this is something that have been proposed by Jacques Monod. The fact that we could model living system using Boolean networks, and more recently, early 2000, the fact that we could model a biological system using what. So, long story short, this is what I want to present here. So what is a metabolic network? So if you sequence genes, if you take all the DNA from one organism, I pass the pipeline and the complexity, but you could extract what are the genes that belong to this DNA. So, and if you have this kind of knowledge, you know that you have gene 1, gene 2, gene 3, genes, and so on. So this is what I represent here as a blue H. No, sorry, yellow square. But because we have also chemical knowledge, you know that these genes, they could be, they could propose, I mean, they could be transcribed into RNA, and this RNA will become proteins. And sometimes some proteins, they are enzymes. And so, for instance, in this particular case, you know that you have, for instance, gene G4. So I know this is remote, but G4 is on the top left. You know that G4 encodes for one particular protein, which is an enzyme, enzyme 4. And because we have beautiful chemical databases, you know that these enzymes is a catalyzer of one reaction, reaction 4. And this reactions is taking one substrate, which is, for instance, component C4, that will become, thanks to this reaction, the component C5 and C5 prime. So basically, I will be very short on this, but if you have the set of genes, some of the genes, they are enzymes. And so you know that because the enzymes they are present within the one particular cell, one particular organisms, you know that potentially we have a metabolic machinery, so which is here just a set of reactions that connect one substrate to another and so on. So the way biologists represent this kind of metabolic network is more or less complicated. So here is one of the cartoons that you could use. This is the metabolic metro map. People in biology, they try to put all the metabolic networks, they try to split it into pathway. So you heard about maybe the carbon fixation pathway, respiration pathway, whatever. And so all these pathways, they're all connected to each other, and they are described by set of reactions. But so, computationally speaking, if you take this kind of graph, so bipartite graph, I call it bipartite because it's a way to connect one component to another via a given reaction. I will bring you back to the first year at the university of or high school, where basically one reaction, you could represent one reaction by the set of components and the stoichiometry associated to the component to be used. For instance, I was talking about reaction 3. Let's say that you know that reaction 3, you have to take 2 times the component C3, 1 time the component C6, and you will be able to create the component C4 and C4 prime with this particular stoichiometry. So thanks to mass action law, you could equilibrate these particular reactions. And so what I propose to do is to project this reaction as a column in a matrix. So basically, you could see that on the right, you have a column R3. You will have -2 in the front of the line C3, -1 in the front of the line C6, and +1 in the front of the line C4 and C4 prime. So, and quickly, basically, you could do this for all reactions. Some reactions, they are reversible. Some reactions are irreversible. Okay, so you could describe all this kind of knowledge into a big matrix. This is something that we call the stoichiometric matrix. So long story short, again, more or less in one given organisms, you could go from 2,000 to 6,000 metabolic reactions. So we have, in this case, a lot of columns and a lot of rows, but also a lot of zeros. This is something to keep in mind, because sometimes, I mean, obviously you don't use all one metabolite in all reactions. What we are interested in is to understand what are the fluxes that will go to these particular reactions, because we could have a flux that goes in one given reaction. So let's say that here I call the flux V, and so we have a flux vector. And basically, if you're a physicist and you want to model the biological system, what you want to do is to assume that, for instance, if you have the variation of one metabolite, the variation of one metabolite is subject to the fluxes that goes to, for instance, one reactions, and the fluxes that goes to another reactions. So one positive effect of the metabolite's variation and some negative effect of the metabolite variations. And basically, you put some weights that are associated to the stoichiometry. So you could generalize this problem. And interestingly to us, basically at the end, you could end up with one simple linear algebra problem, because you know the stoichiometric matrix, you don't know the flux vectors values, but you assume that the whole system will be at steady state. So it means that you have to solve this simple problem, which is S per V equal to 0. On top of that, we could have an additional constraints. Some constraints are associated to the fluxes because for instance, you know that some fluxes, they are always positive. So you know the lower bound of the fluxes. And sometimes you know the upper bound of the fluxes because you know that one fluxes cannot go higher than one particular fluxes. So if you put all these constraints together, basically, you will end up with a solution space. Okay, so one solution space is an n-dimensional space. So you know that any point within the volume or even at the surface of this dimensional space is a solution that is feasible, biologically speaking. And so in a nutshell, I was just presenting to you something that what we call a constraint-based method. So this is something that is very well used in biotech companies. So you have this set of linear constraints. And so you could have a solution space. So here I just project the solution space in 2D. So considering one fluxes in reaction 2, V2, one fluxes to reaction 1, V1. And so in yellow, you have all the solution space that are feasible for these two conditions, two reactions, sorry. On top of that, what people want to do for the sake of biological predictions, you want to maximize a given objective. So for instance, all the fluxes are not equivalent. Some fluxes, they could provide more fluxes towards, for instance, the fat, the proteins that constitute the organisms. So for instance, you could assume here that you want to maximize, you want to get a solution that maximize the fluxes that are within reaction 1, so V1, and the fluxes that are within reaction 2, so V2. So in this case, you want to have an objective and you want to optimize this objective. So you end up with this set of solutions. So all the red line here are equivalent for the biologist, means that all the fluxes within this space, red space here, they are biologically relevant. So this is something that we call the flux balance analysis. Just to show you that the reason why this is interesting, it means that we could also estimate what are the maximum of the fluxes for V2, what is the minimum value for V2, and then you have this range of value. And so it means that all the fluxes between this max and min for V2, they are equivalent and the same for V1. So you could do this for all reactions, which is a way to represent the fact that thanks to just to the knowledge of presence of genes and associated reactions, you could have an estimation of all the fluxes that are feasible for the biological system to live in. So again, that was very description in a nutshell. So to solve this problem, we are using solver like Cplex or relative. This is very well known in biotech. There is a huge amount of paper in the literature that are using this kind of approach to understand, for instance, microbes to improve I would say the yeast production for the beer, for the wine, the yogurt, whatever you name, all the biotech that you want, even recently the oil extractions from bacteria. So this is something that have been very well used for the biotech, but not very much for ecology. And so this is the point of this talk, is to show you that maybe we could adapt this kind of approaches to represent the living system thanks to these descriptions of set of genes, and try to see how we could extrapolate something. So I just want to present to you a manuscript that you could track if you want in ecology letters, in which basically we just project this way of thinking about a biological system into ecology. And what we want to do is to use the concept of niche. So maybe you guys have heard about this seminal concept from Eveillard Hutchinson. This is basically, Hutchinson was a theoretical ecologist, and he was proposing the concept of niche, which is the space in which one organism could live. So the space is something that was at the time very broad, actually. That was just the fact that how much resources are necessary for one organism to survive, okay? So you could have different kind of resource. So for instance, here I just present the different panda bears. So some are using a lot of resource like bamboos or whatever. Some are using smallest amount of bamboos, and so you could understand it. So basically, if there is no bamboo, you die, okay? but if there is more, there is not an effect, and so something else is popping up. So what we did in this concept is basically Hutchinson is proposing an n-dimensional space, which is basically the space in which organisms could survive. And the only thing that matters to draw and to design this space is to consider the amount of nutrients that are associated and also the growth rate that are associated to this. So if you could follow me, this is maybe something that you could relate to what I was presenting just before. So we have a set of genes, okay? From the set of genes, you could have the set of reactions. And so you start from I don't know if you see my mouse, but anyway. You start from the top left, you have the chromosomes, you have all the genes. If you look in the middle top, you have like a metabolic map or what's supposed to go on. So you know all the nutrients that are supposed to be used, because if they are not used, actually the reaction is not supposed to work. And we have an objective function, and the objective function is one, I would say, one particular reactions, one reaction that is not necessarily chemically speaking, but more biologically speaking, what we call the biomass reaction. So like a reaction that take several substrates from the organisms, and you want to maximize the production of them. So we have the solution space on the top right, and basically what the biologists are doing is they try to maximize the biomass, and by taking, for instance, just this red dot on the top of the solution space. This is what they are looking for. If you change gear and then you want to use the concept of niche, basically what we have to do is just to consider the nutrients uptake and productions and the growth rate, and then basically focusing just on this specific space, which is all the space that is above a specific growth rate. This is something that we call the death rate. So the system is supposed to grow higher than the minimum value, otherwise you die. And so Hutchinson was proposing this on formalization. What we propose to do is actually taking the formalization of metabolic network and try to see if it makes sense. So for the few next slides, it will be very biologically driven, but this is just a way to convince you guys that actually this is something that is working. So we are a group of bioinformatics. So the first thing that we do as a bioinformaticians, we try to apply this on one given bacteria, which is E. coli. So this is the most well-known bacteria in the world. That's the best way to test it if it's working, because we have all the knowledge of genes, metabolic networks, everything is there. And so what I present to you is one illustration of the solution space, which is the niche, metabolic niche that you could get from the metabolic network of E. coli. So on the left diagram, you have the exchange of CO2 of this organism. So you could see that the CO2 could be positive, so you produce CO2, but it could be also negative, like you are doing kind of fermentation, okay? So you catch the CO2. And for the x-axis, you could have the exchange of oxygen. So exchange of oxygen is always negative, so you consume oxygen because E. coli is a heterotrophic bacteria. So if you look at this shape from the top, this is what you have on the right. So you could see that this shape is much more complex than you could imagine. This is not just a small pyramid. This is something much more complex. And we represent ages to represent this shape. So this is a beautiful mathematical game of topology. You want to extract this shape and you want to understand what's going on from this shape. And interestingly, if you take the classical niche, the one that people are using in ecology, the niche for ecologists, this is the red square. So this is the maximum value used of CO2, the minimum value used of CO2. And so you have a surface that is much larger than the one that you could use if you consider the genes. And also interestingly, from this shape, you could extrapolate what we call metabolic traits. Because for instance, if I take the green area, this is the area where the system is breathing, so taking oxygen enough. So and if you have the yellow one, basically, this is where the system is making a fermentation. So this is the respiration is quite slow, but you consume CO2 to compensate, I would say, the unavailability of oxygen. So that's something that you could use to design traits. So this is important, you will see later in the ocean, because sometimes people, they consider two different kinds of equations to consider one organism is when there is high oxygen and one set of equation when the system is in low oxygen conditions. So with this, basically, you could encapsulate the full behavior of the system just by considering the full set of genes. For the following, so this diagram, I will be very quick because you guys are not necessarily into biology so much, but on the left panel, we compare what we call the niche that we compute and the niche from ecological niche, actually the one, so the red square I was just presenting. And so all the dots here in this diagram, they represent couples of bacteria. So either bacteria that are coming just from aquatic systems, so this is the blue dot, or they are just coming from the gut microbiome. So this is all the bacteria that you could have within your intestine. So this is the red one. Or you could have bacteria that are coming just from the soil. So here is in brown. And so the gray one represents bacteria that are not necessarily coming from the same. So interestingly, you see that all the dots, they stay in this diagonal, so which is great for us. And most of the dots that go above the diagonal. So it means that we have better precision using genes than just considering the general range of value in which one organism could survive. To us, the two most interesting diagrams were the one from the middle and the one from the right. The one from the middle, we try to learn if there is a relationship between the size of the niche space and the number of reactions that you have within the system. So it means that there is no relationship at all. It's not because you have more reactions within one bacteria that actually the bacteria is just show a bigger solution space, so bigger niche. So, and it's not because you have only one reactions of different that the system will be very close. So this is interesting. If you just consider the number of genes, the number of reactions, this is not a good proxy of the phenotype, I would say. So phenotype means the way the system is supposed to behave. And if you do this and you compare this to the phylogeny, which is on the right, again, we do not have any relationships. So the phylogeny is not associated at all to the potential niche of the organisms. You could have two bacteria that are cousins and are not doing the same at all. So we have to be careful with the phylogeny as well. Sorry, right, about that. So we did the same analysis for the full set of bacteria that have been found within entire ocean. So within entire ocean, we have couple of I forgot, but more we use 5,000 different bacteria, and so we have the same behavior in the marine system. So I'm going slowly to the marine system. Don't be afraid. And then finally, I just compare the niche, the size of the niche, because we have the volume, we could see if one volume is encapsulated into another one. And so we could have like this Russian doll effect of all the bacteria. So we have in this diagram all the bacteria, and you have the size of the nodes that are proportional to the volume associated to the niche that we could compute. And so on the top of the diagram, you could see that we have significantly larger nodes than the one that are at the bottom. And to us, that was very interesting because the one from the bottom, they are more or less bacteria that are, I would say, parasitic. So the more parasitic you are, and the more smaller the niche is pretty small. So you're not supposed to adapt to broad conditions. You're supposed to be very specific. To us, that was very interesting. And sorry, I forgot. And every time you have a line between two dots means that the one from the bottom is included into the one from the top. So that's a way to check the niche overlap just by using again the gene content of each organisms. And I promise this is the last biological slide. And interestingly, we could also check what are the most important reactions to sustain this particular volume. So long story short, again, not all reactions are equivalent. Some reactions are very important to sustain this volume. And so we project this knowledge on the chromosomes of, for instance, here, one diatom. So we have one diatom. This diatom is constituted of 33 different chromosomes. So this is all the lines that I represent here. So you have the chromosome 1, which is the longest one, and so on. So that's the way biologists, they represent their chromosomes. And we put in the gray small histogram the region that are potentially important to sustain the niche. So that was something interesting for the molecular biologists, because using, again, this volume, we could extrapolate where are located the most important sections of the chromosomes, because some chromosomes, part of the chromosomes, they are very sensitive. They are the ones that are here in gray. So to us, this is something interesting to project molecular knowledge to the next level. And so the next level to me is actually using this concept. So that's why I was using, I took my time to explain to you this concept. You have to realize that for most of the biologists, that was kind of a validation. Okay, so that we have now a way to represent omics knowledge into this volume. Okay, and this volume encapsulates all the genes that you could have from one given organisms. And so what I propose to do now is to go one step beyond, is to consider ocean, global ocean modeling. And so, if I may, this is a very old cartoon that was proposed by Mick Follows and Steph Dukevich in 2001. They were showing how you incorporate biology into Earth system model. And the way they were doing it, basically, a long time ago, was using the amount of nitrogen, the amount of carbon, the amount of phosphate, and you put it within equation. And actually, the equation that we are using, most of the time, they are mono equation. So this is a mono system in which you consider the amount of carbon, the amount of phosphate and nitrogen, and then you estimate the growth rate of these organisms based on the availability of nutrients. You incorporate more complexity, you consider quota within the plankton, for instance. So quota of carbon, quota of nitrogen, quota of phosphate. So this is the drop model that people are using. And the more it goes, and the more people incorporate more, I would say, quota. So protein quota, lipid quota, chlorophyll, all these components that are necessary to represent and to mimic the plankton growth. So with this, basically, you had a lot of complexity within the equations that you are supposed to use to represent the metabolic behaviors. And so in the front of this complexity, what we propose to do is to do not a complexification, but this is a mathematical word joke around it. It's using a simplexification, because I was presenting to you the metabolic networks, right? From the metabolic networks, I could extrapolate the metabolic niche. This is I was presenting to you guys just before, but the shape, the mathematical shape of the solution space, this is a simplex. So what we propose to do is to do a simplification of this. So, and to see how we could meet in between. Except what that we propose is pretty simple. It's like, let's say that you have one given organisms, okay? So one plankton, you know this is genomes, you know the genome, so you know potentially is metabolic networks. If you have the metabolic network, we could extrapolate the metabolic niche, which is again the solution space above the death rate. And then we have additional knowledge. The knowledge that we have is like, for instance, in one particular point in the ocean, we know how much nutrients are available. So this is what we call cutting the cake. Basically, you know that we have the solutions, and so you know that you have no more than, I don't know, this amount of nutrients, this amount of nitrate. So you cut it. You cannot have this shape that is higher than this amount of nitrate. And so basically, at each point that you are located to simulate what's going on, you just cut the solution space that we have. So you just cut the metabolic niche that you pre-compute. So, and cutting, this is something that is pretty fast. I mean, computationally speaking, this is something that is easy to handle. And so with this, we compare the growth rate for one given bacteria. So this is a photosynthetic bacteria, Prochlorococcus med4. We compare the growth rate. So we have this metabolic network. And so here on the left diagram, you have a map in which we compare the growth rate estimated using ordinary differential equations, so very complex equations that have been involved into the PSKS model. So on the top, you have the simulations that you could have for the whole set of cyanobacteria using ordinary differential equations. And at the bottom, you have the simulations that we could have using the metabolic network abstraction that I was presenting to you. So the niche and cutting the cake of the niche, I would say, to compute everything. So, okay. this is not striking, but you have a pattern that are quite similar. And so to show you as better illustration, you could have a transect that goes along the Atlantic from the north to the south. And so you could check what's happened at different depths. And so as you could see, you have on the left panel, the color bar, they represent the simulations as proposed by Pisces for cyanobacteria. And the dashed line, they represent the simulations that we have for the genome scale model, so GSM, metabolic model that you could have to represent one cyanobacteria. So we have similar qualitative patterns, even if you do not fit the same amplitude, I would say the same magnitude of the symptom. If you compare the full ocean for all the grids over a year, so we have a pretty good fitting between Prochlorococcus growth rate and the picophytoplankton growth rate. So we have a R2 of 0.8 and the slope is equal to 1.50, which means that actually the prediction is very good actually. But we still have the problem of magnitudes. But the problem of magnitudes, to me, I mean to us, this is something that is not strange, because actually we focus only on one strain of cyanobacteria versus the model of Pisces is taking the full set of picophytoplankton. So we have a different species that are involved. And so what is the interest? It's not just mimicking the picophytoplankton. The interest is actually you could check all the reactions that you want from the bacteria, because, okay, we mimic a bit equations that have been proposed by modelers, but we could also estimate what is, for instance, all the fluxes that goes to the lipids. So this is what I represent on the map here on the top right. With the lipids, we could estimate how much lipids are supposed to be produced to be able to fit the growth that have been mimicking the Pisces simulations. You could do the same for glycogen. You could do the same basically for all the metabolites that you like. And so to us, that was something intriguing, because then we could check, you put it in a PCA, so you have all the variation of environmental parameters here in green, so DOC, iron, temperature, the lights, the oxygen, ammonium, so all the metabolites that are necessary for the cyanobacteria to grow. And then after, in blue, you could have the fluxes of proteins, amino acids, cell wall, pyruvate, glycogen, whatever you want. And so with this, you could extrapolate what are potentially how the system is acclimated to the full system, to the change of nutrients. On top of that, you could also compute what we call the constraints, because potentially the system is more or less stressed, I would say. It means that what is the freedom degree that you have to change the fluxes? Sometimes you do not have any freedom degree, so you're highly constrained. Okay, so for instance, for phosphorus, you could be very, very constrained. And so this is what we present here. It's like if you take for Prochlorococcus, you have the amount of constraints that you have on phosphorus. When it's pretty dark, it means that the system is so constrained that actually phosphorus is becoming limitant. You could do the same for nitrogen. So it's very, to me, interesting, because that's a way to represent the stress in another way. It's like you could take whatever you want, you're not stressed at all. So the constraints are pretty low. So here the colors in the maps are pretty light. But when the stress is pretty high, it means that you have to take this specific amount of here, phosphorus or nitrogen, because if you do not take this specific amount of phosphorus or nitrogen, actually you will never reach the growth rate that you want to reach. So basically that's a way to show the limiting factor and co-limiting factor to whatever you want. And actually this measurements fit is coherent with have been shown actually in different provinces for co-limitation between low P to N versus high P to N ratio. And actually, this is something that have been shown with data sets. So we fit pretty much parameters, I mean, observations that have been made in the ocean. And so behind this fact that you are more or less constrained, we could learn the different acclimation strategies of this bacteria. So for instance, again, I just present here one particular acclimation is the light, not the light, the temperature. So sometimes you could have pretty low temperature. And actually, using this approach, we found that the more you put the system in the cold, and the more you're supposed to store carbon into lipids. So you increase the ratio lipid to biomass ratio. So the colder it is, and the more you stock lipids. So this is something that has been shown in the literature in previous experiments. But this is to us very interesting because this trait, this acclimation trait, this is something that has been, I would say, encoded within the genomes. So from the genomes, we could extrapolate this kind of processes. And similarly, we could do the same using lights, and you check the how you allocate carbon to biomass. So there is several ways to allocate carbon to biomass. Basically, you could compute the glycogen per biomass that you compute, but the glycogen storage index is something that could be interesting because we could see that actually. So I will go quickly, but when there is no light, actually, the system is growing on glycogen. It's like it's not purely mixotrophy, but this is something that look like that we have a way to see that the system actually it could grow to something that light. It could grow potentially to glycogen. So if you have a highlight, basically you build the glycogen stock. This is what we have on the diagram lower to the right. And when there is no light, you use the glycogen stock to survive. So again, this is something that we could use. This is something that we could provide to the modelers without no parameterization at all, just because it's encrypted within the genome. So if you follow me, just the last results that I want to show to you guys is the fact that because we have using something genome scale, because I hope I convinced you guys that the traits are encoded within the genome, you could check the fluxes of glycogen, lipids, or whatever, but you could also check all the metabolites that we want. And that was a review that have been proposed, I think it was 2 years ago now, about all the microbial metabolites that could be used in the marine carbon cycle, in particular to what are the metabolites that are constitutive of the DOC, so dissolved organic carbon. And so we could propose this kind of maps. And this kind of maps, they are never been used, never been proposed so far. In particular, we compare two different organisms. On the left, we have Prochlorococcus, so cyanobacteria. On the right, we have a diatom. And so for each of these organisms, we could have a map of production of metabolites. So for instance, it has been shown that Prochlorococcus could provide 33 metabolites that could belong to the DOC, versus the diatom on the right could provide 19 different metabolites to the DOC. And so with this, we could check the maps, provide some maps in January or in July across the global ocean, for which we could estimate the intensity of DOC productions. So when it's pretty dark green, it means that we have a high productivity of DOC. So as you could see, we have a different pattern between Prochlorococcus and so the cyanobacteria and the diatom. So the diatom produces much more DOC than cyanobacteria. But on the top of that, we could also estimate what is the diversity of molecules, the diversity of metabolites that are associated to this DOC. And so what we mean by diversity, you could have a high diversity, which means that you have a lot of different metabolites that have been proposed, versus a low diversity, in which case you have only, for instance, one metabolite that have been proposed to make the DOC happening. And to us, that was very interesting to see, for instance, that the diversity associated to the Prochlorococcus, the cyanobacteria, is kind of different patterns than the diversity that you could see from the diatom. And so, as you could interpret it, is basically the high productivity of DOC is not necessarily associated to the high diversity of the DOC. So to us, that was intriguing because in this way, we could mix a bit the different problems like IPCC problem, which is just the DOC productions versus diversity associated to this diversity, so IPPBS. We have from this diagram different provinces, more or less provinces driven by diatom for the DOC production versus provinces driven by cyanobacteria. And interestingly, we could see that basically the cyanobacteria, they produce a lot of DOC hotspot, but within this spot, basically the diversity is much smaller than the spot that you have for the DOC, which means that the cyanobacteria, they are more or less free rider. They don't give much to the, they don't give much metabolites to the community. They only give particular metabolites, which is the opposite for the diatom. The diatom, as you could see from the map, the blue map is much larger. And so Thalassiosira, in this case, is providing a lot of different flavor, I would say, in the ocean. And that's something that provides a lot of different DOC. So potentially, this is something that could provide more trophic systems and providing more materials to the trophic system. So I'm almost there. So as a perspective, basically the plan was to incorporate this omics knowledge. So omics is again the set of reactions, set of genes. So we have all this kind of knowledge that is coming. The host system model, I think now we could track more than 10,000 reactions. So this is something that is actually feasible. We are ready to incorporate more community, more diversity of the community. And actually, this is something that we could use to track potentially what are the essential genes that we need to monitor, because some of these, not all these genes are equivalent. And so maybe we could use these genes as a proxy and to help, for instance, in BGC Argo, I know that Hervé Claus did a seminar in this same panel, but we could provide new genes that could be potentially new biomarker that maybe you could track. And with these particular genes, they could represent potentially acclimation strategy, all this kind of stuff that could be used. And finally, this is a long shot, but we could also check what is the different evolution scenarios that we could tackle. For instance, what's happened if I shoot down one particular genes, mutation or whatever. And so if you shoot down some genes, you could see the consequences at the global I would say, distribution of organisms and growth rate of organisms. And I will stop here. Thank you.
Well, thanks a lot, Damien, for the presentation. I hope you can hear me. Yeah, it's okay?
Yeah.
Okay.
I don't see you. I need to switch to my screen.
I think it's better now. It's always nice also to hear from works that we are not always familiar with, I guess, for a lot of people in the audience. We have time for some questions. I don't see any in the chat, but if some people in the audience have questions, and if not, I have some. Maybe you're afraid a bit, the people, Damien, are not familiar with the topic, but maybe I can start. So yeah, I'm really not a specialist of this topic, so I learned a lot of things. And looking at the beginning of your presentation, as you know, we are mostly familiar with AI and machine learning methods. And you talked about some words that sounds to our ears just like metabolic network, metro map. I heard the word of graph when you talked about the flux vector in steady states. And I'm wondering, it's very general questions, but if this AI community is bridging with yours in the last years, and if there is some interesting works that you have already identified to help you in your modeling.
Actually, so yes and no. So that's a very weird answer. So if you take the history of AI, the first solver that have been used, so logic programming and constraints programming, they were the first origins of AI. So we are just using the old tool. Yeah. So the solvers, they are up to date. So the solvers are getting better and better. So that's why we are able to solve this problem now. So to be honest, we are using the state-of-the-art Cplex solver for linear algebra and stuff like that. I didn't present the detail because I think that was not the point. That was more the point to show you guys that maybe we could do something with these tools. But It is true that so in my lab, so we are a computer science lab. So S2N is a computer science lab. So there are teams dedicated to designing new solver, and we are using this kind of solver actually to compute populations dynamics and stuff like that. So we are using. So we spend more of time to in the AI framework. We spend more of our time to design the problem in a way that we could fit this question to solvers than actually designing new solvers. But I have to agree that for some questions that I was not presenting here, the solvers are not good enough. And so we have to work with the team around us. So I would say we formulate the problem in a way that is realistic for the biologist and realistic for the solvers that have been state-of-the-art solvers so far. Okay. And so the high I would say we are not, so you could do some machine learnings and stuff like that, but actually we are within the frame of logic programming and constraints programming.
Okay, because maybe one thing I didn't understand quite well, again, in this flux vector in steady state, you show us some equation. Mm-hmm. If I followed you, it comes from the metabolic network graph, right?
Yep.
And I'm not sure, is it in very high dimensional space? Because if it's the case, I guess the solver can be quite long or not.
It is, but it is a very high dimensional space. So the n in this case, n is equal to 5,000. So we have 5,000 dimension. And within this space, what is very helpful for us is like we have a lot of zero. So basically solving this kind of problem is kind of easy. So to us, learning the solution space for one given plankton. So if you give me one genome, from the genome we extrapolate the niche. Yeah. It took us like 10 minutes to compute the solution space. And then after it's linear when you project it into the global ocean, it's linear in computing time. So it's very easy and short to compute. So yeah.
Also, maybe, so you presented us some examples of metabolic network graph. It comes from, how can I say that? I don't know, human knowledge, I mean. And
it's not human knowledge. It's actually from the set of genes. You could build up this kind of graph. So do we have like learning techniques that you could use for that?
Okay. And is there still some relationships that are not identified in this network, so that, I don't know, maybe again modern AI and learning-based methods can be helpful to identify new connections, etc.
Yeah, because this is something that I put under the carpet. But actually, if you check the set of genes, actually we know that I would say in the ocean, actually 70% of the genes are unknown. So they are genes, but we don't know what they are doing. So actually, you're right. We build up on the current knowledge. So we have chemical databases, we have stuff like that. But most of the genes, they remain unknown. So and so you could have a complexification of the metabolic network that will come in the future, but the plan to us is more to merge this community effort, which is the biological community effort, towards the Earth system ocean effort, just to bring the biology, I would say, in the system. Because to be honest, the current Earth system model, they use biology, but they have just few equations to represent life.
Yeah.
So if you take Pisces, so Pisces is beautiful. Don't tell me, no, I don't want to be offensive or whatever. Pisces is awesome. They are doing a great job, but they just have few equations to resume the life. And in this case, we do not have any information about what are like we know that plankton will acclimate to climate change. So we know that some genes will pop up. We know that some genes will be deleted. But with the current framework, with just a set of equations, we have no clue how to incorporate this kind of mutations. And so that was the plan, just to be able to mimic the growth rates that have been parameterized by decades of effort, and try to see that, okay, we are not that far. And because we are not that far, then we could simulate what are the consequences of having some mutations or whatever. because we know that, for instance, if you have this process of this is well known. We have Atlantification of the polar system, so we know that the system, the polar system, is getting more and more Atlantic. But if you check the genes are not the same. So the phenotype is getting the same, but the genes are not the same. So we have no clue exactly what are the consequences of these processes. So that was, to me, it's a long shot, okay? But the plan is to better incorporate this genomic knowledge, because this is actually now the state-of-the-art knowledge from the biological side, and try to see how we could bridge it towards Earth system model, and we could help parameterization also as well, because we know that we have several traits. So how many equations do you need to better parameterize?
Okay,
it's a dream, but yeah, that was the goal. So and again, going back to the computation and the modeling and AI stuff, we are just using the AI as a way to abstract one knowledge into another abstraction. And because we have another abstraction, this is something that is feasible for physicists and Earth system models. So that's how to transform one knowledge with it, which is in one abstraction set of genes, to put it in a form that is more tractable to physicists and biogeochemists. So that's just a start.
Okay. So I think it's a good transition to my last questions, because you just discussed about what's actually done in a system model to model life, and you showed us some maps of growth rates. I don't remember which slide it is, but
yeah, sure.
Where you identified similar patterns from. So if I understood you well, ODE-based set of equations that are actually solved in a system model, and the outputs from a genetic model. And you said that the patterns are quite similar. So I see that. And also that there is some difference in terms of magnitude of the signal.
Yeah.
And do we have some validation with which one is good? Do we have some information about that?
So we have kind of validation. The thing is, so we are facing something complicated. So because the way usually you validate the Earth system model is by using counting particles. So you count cyanobacteria. So basically, in this case, you count how many cyanobacteria you have. So that's a way to parameterize it. The problematic that we have here is we identify not the big group of cyanobacteria, but one specific strain of the cyanobacteria, which is here MED4. So it's very complicated to have quantitative measurements of this bacteria, because when you do some countings, you put, for instance, one marker on the chlorophyll. This is what people are usually doing. So you count how many dots do you have, and that's a way to estimate the amount of plankton that you have in one particular location. This is not something feasible using DNA, actually, because it's a long story, but the DNA, you do not have quantitative measurements. You only have semi-quantitative measurements, because you cannot normalize. So for physicists, there is no unit in biology.
Yeah. Okay.
There is no unit of genes.
Okay.
You could just say that I have 10 times more genes in this location than in this location. So we just have qualitative validations. So qualitatively speaking, we fit pretty much. So that's why this is the best so far that we could do. But the further validation that we have is actually the rates. That's what I was showing about other measurements, in particular when I compare the P2N, and we could predict something. The bioprovinces, they are quite equivalent. So this is more or less the validation that we could have. And it's a long shot, because actually to be able to target one particular species, you have to make molecular stuff that are not available so far. Quantitatively speaking. So there is some techniques, but spiking or whatever, but I think it's too complex to discuss here.
Okay, but at least that was a good start. I don't know if anybody else has some questions. We reached our 1 hour time slot. Okay, I think that's all. Maybe, Damien, I didn't ask you at the beginning, but I recorded the session.
okay.
Is it okay? Is it okay
Sure.
for you to publish the video on our website? And maybe also if you can transfer me the slides, if that's okay,
Sure.
of course. And
sure.
I can send a last email regarding this webinar so that people interested in can have a look at it. Well, okay. Thanks again, Damien, for this very interesting talk. And I hope I'll meet you next time, maybe in Brest, in a transdisciplinary sessions, maybe. So let's do that, and have a nice day, everyone. Thanks again.
Thank you.
Bye-bye.
Bye.